Biology part 190. Well, this treatment exposes all parts of the seedlings equally to the gravitational stimulation, with the result that the radicals continue to grow horizontally. For many years it was believed that the geotropism could be explained by the redistribution of the auxin in exactly the same way as that of the phototropism. This is still thought to be the correct explanation for negative geotropism in shoots, and there is good evidence for the movement of auxin to the coleoptiles in the response of the gravity. However, auxin does not seem to be responsible for the positive geotropism of the roots. It has been that a different hormones, that is called abscisic acid, ABA, accumulates on the downward sides of the roots, and it is hormone which is now thought to be inhibit of the elongation, while the cells in the upper regions continue to grow, producing a downward curvature. The mechanism used by the roots and the shoots to detect the gravity appears to involve all radicals and the amyloplast containing starch grains, and these have a density greater than that of the cytoplasm and have shown to sink under the effects of the gravity. Coming to rest against the cell membrane at the bottom of the cell, referred to the figure, the presence of the amyloplast is thought to trigger the transfer of the auxin and the abscess across the membrane. Similar activity in such cells results in the redistribution of these hormones to the lowest regions of the root or shoot. Cells which contain amyloplast and which seem to be specialized in the gravity detector are called statocytes and have been discovered in all plant organs capable of responding to the gravity. Apical dominance. In most plants, the growing tip of the stem inhibits the growth of the lateral buds and delays the development of the lateral branches. This effect is called the apical dominance and involves the production of auxin by the apical meristem within the apical bud. Sometimes the inhibition produced by the apical bud is weak, giving a plant with a wide branching pattern. In other species, the apical bud has a strong inhibiting effect and plants become tall and thin with little lateral branching. The mechanism of apical dominance is not fully understood. The effect of the apical bud can be mimicked by replacing the apical bud with the auxin and the lanolin paste, but it's clear that the other plants' hormones, including the abscisic acid and the cytokinase, also play a part. In some studies it has been found that auxin would support the only if the lateral buds had not been allowed to stop growing. Once sprouting the lateral buds had taken place, auxin supplied at the tip did not arrest their development. Additional plant hormones. The main types of the plant hormones and their important functions are listed in the table. This section describes the different groups of the hormones and comments on the interaction which produce the particular plant. First of all, let's talk about the gibberellins. These were first isolated from the parasitic fungus, the Gibberella fungicori, which causes the damaging disease of rice. Infected seedlings grow abnormally tall and their stems are weakened. The fungal extracts yielded a number of substances, one of which was named as gibberellic acid. Analysis of plant tissues by the same methods soon revealed that the smaller amounts of the gibberellic acid and related compounds were normally present, and led to the discovery of their role as plants' hormones. At least 50 different naturally occurring gibberellins have been identified. The gibberellins stimulated the elongation of the cells in the stem, are known to be deficient in some dwarf varieties. They are also involved in the leaf growth, fruit development, germination, flowering. In germinating barley seeds, gibberellins produce the embryo plant are known to stimulate the production of analyze enzymes, leading to the mobilization of stored food reserves in the early stages of germination. And we can find them elsewhere as well, and they do provide the evidence for the development. Next we'll talk about number 2, cytokine, number 3, abscisic acid, number 4 ethene, and we'll move on, but right now that's the end of the video.